Okay, so the next one is dihybrid cross. Dihybrid, di means two. So for this case, we have two characteristics that make a cross. Okay, so this is related with the Mendel's second law. Mendel's second law, known as law of independent assortment. Okay, so for dihybrid cross, it is the inheritance of two characteristics, each of them controlled by different gene at different locus on different chromosome. So we look at the diagram here. Okay, for example, we have two different genes. First is the gene to control the color. Either it is yellow color or green color. Second one is the gene to control the seed shape. Either it is round in shape or wrinkle in shape. So we have 1 and 2 characteristics. So this is called dihybrid cross. So for this cross, they control by different gene at different locus. So for different locus, for example here we have first pair of homologous chromosome to control the characteristic of color. Second pair of the homologous chromosome to control the gene of the shape. Okay, this is for the color. They are on different chromosome. Okay, so we have two different pairs of homologous chromosome. First pair and the second pair. Alright, so this is what we call dihybrid cross. If we compare with the monohybrid cross just involve one pair of homologous chromosome with only one characteristic. That is only A, one characteristic. For this one, we have two characteristics. That's why we use two different letters, P and also Q. Okay. So for Mendel's second law, it's called law of independent assortment. So generally, assortment means the arrangement. Okay, arrangement. So it states that each pair of alleles segregate independently of each other pair of alleles during gamete formation. So remember that we have two pairs of alleles. Okay, two pairs. So they say that each pair segregate independently with each other pair of allele during the gamete formation. Okay, maybe it's quite abstract to understand now, so you can look at the diagram to understand better here. Alright, so now for example, in this cell, we have two pairs of homologous chromosome. Okay, the big one and the small one. Okay, so now you look at the diagram here. For the big one, it controls the gene A. The small one, it controls the gene B. So during the stage of metaphase 1, the homologous chromosome align at the metaphase plate. So how they arrange, how they align, or how they assort, it is independently. Independently means that nothing to control them. It is nothing, there's no rules to say that this big A must always align with this big B. It can be the big A aligned with the small b. Okay, so this is, the chance is 50% or 50%. Either you can produce this big A, big B, or you can produce this big A, small b. Alright, so the other chance of getting the gamete here, you have the small a, small b, and the small a, big B. Okay, so the combination of all the possible alleles. So we have 4 different possibilities. Big A, big B, small a, small b, big A, small b, and also small a, big B. So this is what explained by Mendel's second law, independent assortment. We have all different sorts of the gametes. Okay, came as a result from the metaphase 1, how they assort with each other. Right? Okay, so now I will continue to show you the genetic diagram of dihybrid self-cross and dihybrid test-cross. So first we come with dihybrid self-cross. Okay, so first of all, dihybrid self-cross always involve two characteristics. So let us give it a letter, big A. Okay, let's say it represents the tall allele. Small a is the recessive case, so it is the short allele. So another letter, we give it a B here, represent the purple flower allele. Small b represent the white flower allele. Okay, so now every genetic diagram starts with the parents' generation. Okay, so for parents' generation here, what is the genotype of the parents' cross? For self-cross, we must always start from the pure breed. Pure breed means homozygous dominant case. So it is a cross between homozygous dominant case and homozygous recessive case. Big A, big A, big B, small a, small b, small b. So for this case, they are tall and also purple. For this case, they are short and also white. So first, came out with the genotype. Next one must come out with the gametes. So what is the gametes from these parents? Remember the second law of the Mendel say that law of independent assortment. So now this big A pair with the big B, or big A pair with the big B, or big A pair with the big B, or big A pair with the big B. Meaning that for the pure breeding, we have only one possibility of the gamete can become out. Big A, big B. Okay? Remember that it will not come out as the big A, or big B, because it applies the Mendel first law. First law is known as the law of segregation. The big A must segregate from the big A. So you never get the gamete that is big A. Okay, so now this is big A, big B. So for this case, same thing, we have small a, small b. So our F1 generation, all of them, big A, big B, small b, which are tall and purple. Alright, all of them tall and also purple. So now to conduct self-cross, we must have F1 to cross with F1. So big A, big B, small b, cross with big A, big B, small b. Okay, so next one, we come up with the gametes. So for these gametes here, it is very good to apply the law of independent assortment now, where we have already seen in the meiosis diagram, they came out with 4 different possibilities of the gametes. So we have 4 different gametes here, big A, big B, or big A, small b, or small a, big B, or small a, small b. So for this case, also the same thing, big A, big B, or big A, small b, or small a, big B, or small a, small b. Okay, so now it is much more complicated compared to the monohybrid cross that we learned before, because now we have 16 possibilities for your F2 generation. So for this case, we need to construct a Bonnet square. Okay, so I'll teach you how to construct a Bonnet square here. It is make you easier to see the possibilities of your F2 generation. So for Bonnet square, first we draw a table. Okay, on the left and on top we write our gametes. Okay, gamete from the female parents and gamete from the male parents. So all these gamete from the parents here, we put it down. This is big A, big B. This is big A, small b. This is small a, big B. This is small a, small b. While here also the same thing, big A, big B, big A, small b, small a, big B, and also small a, small b. So for here, what is inside the box or the table, you just combine everything. Big A, big A, big B, big B. Okay, you just arrange A with A, B with B. Here is the big A, big B, small b, big A, small a, big B, big B, big A, small a, big B, small b, big A, big B, small b, big A, small b, small b, big A, small a, big B, small b, big A, small a, small b, small b, big A, small a, big B, big B, big A, small a, big B, small b, small a, big B, big B, small a, big B, small b, small a, small b, small b, small a, big B, small b, big A, small b, small b, and big A, small a, big B, small b. So now we have 16 possibilities of your F2 generation. So if you want to see the ratio, what is the ratio for your F2 generation? It's very easy to remember. We have 9, tall, and purple. Okay, this is your not genotype, phenotype ratio. Okay, phenotype ratio means something you can see. So now tall purple, which one is the tall purple? You can see from here, this one. Oops. Okay, this one, this one, this one, this one, this one, this one, this, 1, 2, 3, 4, 5, 6, 7, 8, 9. Is something wrong here? 1, 2, 3, 4, 5, 6, 8, 9. Okay, this one is not. Okay, this one is not. Alright, 2. How many here? It is 3 tall, white, 2, 3 short, purple, 2, 1 short, white. Okay, 1 short white is referring to our this one. Okay, so our ratio, very easy to remember the number here, 9, 3, 3, 1. And this is for phenotype ratio. What about the genotype ratio? Genotype ratio, usually we do not state it. Genotype ratio, because it's too much. Like how many of them big A, big A, big B, big B, how many of them big A, big B, small b. So we usually don't say that.